Hi everybody, my name is Bao Nguyen, and I will present to all of you my summer research in the tandem mass spectrometry of isomeric polycyclic aromatic hydrocarbon derivatives. Let's start from the building blocks. Hydrocarbons are simply compounds that are made up of hydrogen and carbon atoms. Benzene, or C6H6, is an example of a hydrocarbon. By shorthand notation, benzene can be also represented by a hexagonal ring. A benzene ring is also referred to as an aromatic ring. When multiple aromatic rings are fused into each other, they make up polycyclic aromatic hydrocarbons, such as anthracene with 3 rings. My research focuses on the compounds derived from anthracene and their isomers. The anthracene derivatives are connected to a carbon-nitrogen double bond. So, what are isomers? Isomers are compounds that consist of the exact same types and numbers of atoms but have different structures, which affects the chemical properties. As an analogy, the same Lego bricks can be built into different objects, like a gun or a sword. Isomers of anthracene derivatives I research vary in the length and the branching degree of the alkyl chain connected to the nitrogen atom. The isomers also differ in whether they have an attached methyl group or not. As an example, these two molecules are isomers with the same chemical formula C20H21N. The compound on the left has a linear alkyl chain, while the one on the right has a branched alkyl chain and a methyl group. My research in aromatic hydrocarbons is relevant to the field of petroleomics. Aromatic compounds are very valuable in petroleum because they determine the yield in highest octane gasoline. However, distinguishing isomers pose challenges in studying the molecular components in crude oil because isomers share the same chemical formula and molecular mass. Therefore, I've been investigating 3 pairs of isomers to propose strategies that can discriminate those isomers in a mixture solution. To tackle this problem, I've been working with mass spectrometry, an experimental technique that measures the abundance of ions existed in a solution, shown as peaks of corresponding mass-to-charge values on the spectrum. As an analogy, when I run a pack of coins through a mass spectrometer, the produced spectrum will show how many coins of each type I have. However, as I mentioned earlier, isomers share the same mass, so their peaks overlap onto each other, and mass spectrometry alone cannot distinguish them. I had to take one further step to tandem mass spectrometry, or MSMS. Here's another analogy. 2 puzzles created from the exact same pieces but with different arrangements, representing 2 isomers. What MSMS does is that it breaks these puzzles apart into smaller fragments. As you can see, they make different types of fragments. This works for chemical compounds as well. Different isomers have different fragmentation patterns. Studying these fragmentation processes provide clues to distinguish isomers. MSMS experiments start with the ion formation phase, where analytes are introduced into the machine and ionized. Then the quadruple selects certain ions based on a given mass-to-charge value to avoid other contaminants. The selected ions enter the collision cell and counter collision gas, where they are induced to dissociate into smaller fragments if enough collision energy is applied. The precursor ions and their fragments then progress into the flight tube, where they are navigated to the detector by the orthogonal accelerator. Basically, lighter ions take shorter time to reach the detector and vice versa. Based on this principle, the detector can determine the mass-to-charge value of the ions and then measure the abundance of each type of ions. Next, I will present and analyze some results obtained from the research. First, I investigated the isomeric pairs A and A1, which are labels my lab uses for convenient purposes. The IUPAC names can be found below. The ionized molecules are isomers with the chemical formula C21H24N+. A has a longer alkyl chain, while A1 has a shorter alkyl chain and a methyl group attached. From the MSMS spectrum of A at collision energy 25 eV, fragments at mass-to-charge 179, 205, and 206 are detected. From the spectrum of A1 at the same collision energy, fragments at mass-to-charge 205, 220, and 275 are detected. I was seeking for the difference in the fragments produced by A and A1, so mass to charge 205 is ignored. The other fragments in the table are unique to the corresponding isomers. They can be used to identify and distinguish A and A1, so I called them fingerprints. I prefer to use the more abundant ones, so I chose mass to charge 206 and mass to charge 220 to be fingerprints of A and A1, respectively. Then I made mixtures of A and A1 at different proportions and ran the samples through the mass spectrometer to validate if the chosen fingerprints are accurate indicators of A and A1. From the MS data of A and A1 mixtures, higher abundance of mass to charge 206 fragments are detected at a higher percentage of A in the mixture solutions. The correlation is approximately linear, determined by the R-squared value. Likewise, higher abundance of mass to charge 220 fragments are detected at higher percentage of A1 in the mixture solution, which also shows a linear relationship. These results confirm that the chosen fingerprints are accurate indicators of their respective isomers. Next, I investigated isomeric pair L and L1, whose ionized molecules are isomers, with the chemical formula C19H20N+. L has a linear alkyl chain while L1 has a branched alkyl chain. None of them has a methyl group attached. From the MS spectrum of L at collision energy 20 eV, small amount of fragments at mass to charge 179 and 206 are detected. Like L1 also creates the same fragments at mass to charge 179 and 206, so there are no fingerprint fragments, unfortunately. However, it is important to note that L1 produces a significantly larger amount of mass to charge 206 fragments compared to L. To further analyze this difference, I made breakdown graphs that demonstrate the abundance of mass to charge 206 fragments in relation to the collision energy applied. I could clearly observe that L1 molecules produce these fragments at a lower energy level than L. I applied this knowledge to run MS experiments of L-L1 mixtures at different ratios. As the percentage of L1 increases, the graph shifts leftwards and upwards, indicating lower collision energy required to generate mass to charge 206 fragments and higher abundance of these fragments formed. This is consistent with what I found earlier, thus this feature can be used to distinguish L and L1 in a mixture solution. In conclusion, my research proposes two different strategies to differentiate isomers. Isomers with unique fingerprint fragments can be determined based on their fingerprints. In practice, this knowledge can be applied in establishing linear regression models from the MS data of an unknown mixture to solve for the relative concentration of known isomers. When isomers produce the same fragments, the fingerprint method is not feasible. In this situation, collision energy demand to form fragments can be analyzed. I observed that molecules with branched side chains require less energy to dissociate themselves than those with linear alkyl chain. If I had more time, I would like to expand this research to further study how to distinguish more than two isomers, which is the reality in the highly complex crude oil mixture. I am sincerely grateful to all staff members of the STARS program for offering me a valuable opportunity to conduct research within a professional laboratory setting as a high school student. I acknowledge my advisor, Rhonda Wandel, for valuable feedback to this research paper. I would like to express my gratitude to my mentor, Dr. Benjamin Bythell, and all the lab members for their warming support and guidance throughout the research. I would like to extend my special gratitude to my parents who have always wholeheartedly supported me to pursue my science passion and study abroad in America. Last but not least, thank you all of you for being a superb audience. I hope all of you enjoy the rest of the day.